ওয়েট করছেন, আমরা 2 মিনিটের মধ্যে আমাদের কিনোট স্পিচ শুরু করব। থ্যাংক ইউ। স্যার ইজ দেয়ার? জয়েন করবে, আমাকে বলছে স্যার শুনতে পাচ্ছে। Our volunteers are requested to escort all the participants and guests back to the hall. We are going to start our keynote speech within few minutes. Hello. So now this is the time we are going to start the keynote address. The keynote address will be given by one of the most prominent figure in the field of dairy microbiology, and more precisely in starter culture area, and also my guru, Professor Dr. Rameshwar Singh, Honorable Vice Chancellor, Guru Kashi University. Sir, although we are missing you a lot for your physical presence, but thanks to the IT development, we are having you virtually, and the next couple of minutes is assigned for you, sir. So please, you can start. I think Prajapati sir wants to speak something. So after Prajapati sir's address, you please start your keynote speech.
Thank you.
Most welcome, sir. Dr. Rameshwar Singh is one of our most important person on our SASNet Governing Council, and also We are missing you, sir. Uh, but
Same here.
with pleasure to listen to you. So you may please start, and then we are all eager
Okay.
to listen to you. Some of the students are still outside in the tea break. Please join immediately. We will not get such a chance to listen to such a stalwart in the field of dairy microbiology. Most welcome, sir. Please.
Namaskar, Doctor. Ma'am, I hope I am audible. Hello, am I audible?
Yes, sir. Yes, sir. Very clear. Very clear.
Okay, okay, thank you, thank you. I would like to share my screen. At the outset, I greet the chair and all the dignitaries, invited guests, Dr. Prajapati, Honorable Vice Chancellor, all the delegates, distinguished speakers, and my dear students. I am highly thankful to the organizing committee, Dr. Prajapati, Professor T.K. Datta Sahab, and the organizing secretary for inviting me to deliver the keynote address to this important conference. ASNET is a very important platform for discussions on probiotics and taking the cause of gut health and microbiota in general. Its regular meetings are being carried out by Dr. Prajapati and his team, and I always compliment Dr. Prajapati for this. So I also regret that I could not join you physically because of the interruptions in the flights and also the inclement weather due to very dense fog in northern parts of the country. So I would like to share my screen and as a sorry. I have sent a request for screen sharing. You may enable me.
Sir, please allow.
Okay. Okay. Now it is on.
Sir, please allow, sir. Click it, sir.
Yeah.
Yeah. Yes, sir. It is visible, sir.
Yeah, I'm
Make it,
doing
sir,
it in
full
the
screen, sir.
presentation mode. Yeah.
No, no, full screen. Sir, from the beginning, make it is full screen, sir.
Okay. Okay. So I'll first open it and make it.
আচ্ছা ঠিক আছে পরে দেখছি তুই এখন যেটা আছে সেটা পড়ে দে তারপর পরে পাঠাচ্ছি ওইখান থেকে শেয়ার করতে হবে Sir,
Just come in.
okay.
I was equally keen to join all of you, and really missing the opportunity to meeting my old friends and students. So, coming back to the keynote, I will give an overview of the probiotics, starting with introduction, classical functions of probiotics, beneficial metabolites produced by different organisms, and the diverse probiotic strains, new areas which are emerging, new tools which are being used, and the future directions in probiotic research and also the ethical issues. So we all are very familiar with the definition of the probiotics, and we know that WHO and International Society for Probiotics, they have given the definition long back, and they define this living organisms when administered in adequate amounts. So there is a number 10 to the power 6 if you are having a dose of 100 gram, and they should confer health benefits to the host. They are primarily recognized for their positive effects on the gut health, but their influence extends to various bodily systems. There are criteria which are also very well known, and I will just cite some of them for the sake of repetition. These are they must be safe, non-pathogenic, non-toxic, and also free from transferable drug resistance or virulence genes. And strain level identification and stability are important because the benefits are strain specific and not generic to the species. So it is the individual strain which is important, and they must survive in the gut. That is the low pH, presence of bile salts, and anaerobic conditions and other conditions in the gut. And functionally, they should be show the efficacy and produce the beneficial effects. Then only they can classify as probiotics, and clinically proven benefits. That is, they must demonstrate strain-specific health benefits, which can be supported by experimental and human studies. So technological and self-stability, that is also important because the probiotics have to be viable, and they have to withstand the rigors of the environment, that is the processing, storage, and delivery. Commonly used organisms, generally lactobacillus, bifidobacterium, and the Saccharomyces, etc., are common. But there are more organisms which I will talk about during this presentation. However, one thing I wanted to share, especially with the young students, is that extensive taxonomical revision of the genus Lactobacillus and Leuconostocs has taken place in 2020. And out of one genus, Lactobacillus, 23 new genera have been added. And most of the probiotic strains, however, they have been assigned to Lactobacillus, Lactobacillus, Limousin Lactobacillus, Levi Lactobacillus, and Lactobacillus. These upgraded names are now used in scientific communication and increasingly in commercial labeling. If any claim is made or submitted to the regulators for grant of license or approval from the regulatory agencies, then these revised nomenclature, which is valid nomenclature, has to be used, and the guidelines in this regard have been issued by different regulatory agencies. This guideline which I am presenting here is from the government of Canada, and they have said the new name of the strains of Lactobacillus species, and within the parentheses you can give the previous name, and you can refer to the valid names as they are published in the International Code of Nomenclature of Prokaryotes, and you can look at the various resources on the internet which are available. So you can refer to either NCBI or to the valid names of prokaryotes, that is the list of prokaryotic names with standing in nomenclature. This we normally do in microbiology when also we are writing some publications or we are referring to this. So these are some of the changes in the nomenclatures. We all know the Lactobacillus rhamnosus GG, Lactobacillus KGI shirota. Now the names have been changed. So if any work is reported on this Lactobacillus rhamnosus, will now become Lacti-Kezai bacillus rhamnosus, and Lactobacillus kezai shirota remains this, also becomes Lacti-Kezai bacillus. Similarly, Paracasiae, again Lacti-Kezai bacillus Paracasiae. So this shows the changes in the names, and also these are well-known probiotics. So the common functions of these probiotics are also can be seen in this slide, and other organisms also similarly. The nomenclature has been changed for almost all the Lactobacillus strains, except Lactobacillus delbrueckii and Lactobacillus acidophilus. These have been changed. So the original paper from Zhang et al. Can be referred, or many other papers have followed on this taxonomy, and they have decoded it very well to explain the changes. This has been done to accommodate the genetic heterogeneity, because there were 300 species under the genus Lactobacillus, and they were quite diverse. So with the advent of the molecular biology and gene sequencing, the importance of the strain, they have been reclassified, and this change we must accommodate in our teaching as well as in our usage in the research papers and our products. Bifidobacterium, no change, and Saccharomyces also no change. So this shows some of the common and widely used probiotics. Now I come to the classical functions of the probiotics. You all are familiar with these classical functions, and that is the major function which is considered as the well-being of the gut microbiota, that is the maintenance of the balance of different species within the gut, which is so important for digestive health, for the immune functions, and to maintain this microbial balance. The phenomena are also well known, that is the competitive exclusion. They occupy the space in the gut, adhere to the mucosal epithelial lining, and displace the pathogens through their competitive adhesion nature. Then they produce organic acids and also produce antimicrobial substances like bacteriocins, which may inhibit specific pathogens. Then there is barrier protection. They strengthen the intestinal epithelial integrity and do not allow the leakage. Immune modulation is also very well known in the classical functions of the probiotics. Coming to immune regulation, the mechanism is through promoting regulatory T cells, enhancing the production of different cytokines, particularly the anti-inflammatory cytokines like interleukin 10 and TGF beta, or strengthening resistance to pathogens, reducing the risk of allergies and inflammatory diseases. Probiotic organisms such as Akkermansia, Mucinifera, and Faecalibacterium prausnitzii enhance intestinal barrier function by upregulating tight junction proteins, including zonula, occludens 1, occludin, and claudins. when we were doing our regular, curricular studies for masters or PhD, these organisms were not discovered, and now these organisms are treated as one of the most important organisms, that is Faecalibacterium prasunitzi is can be in the range of 5 to 15% in healthy human gut, and it is now very well studied species. Likewise, Akkermansia mucinifera is a beneficial and a mucin degrading bacterium that naturally resides in the mucous layer of the human intestine, and it is also quite abundant, going up to 1 to 4% of the healthy adult microbiota. Then they produce different molecules, different chemicals, which are very helpful, and they can exert the beneficial effect through production of acetate, propionate, and butyrate. And these small chain fatty acids, they cross the blood-brain barrier, they regulate neuroinflammation, they also influence neurotransmitter synthesis, and they modulate microbial functions. Many probiotic organisms are capable of synthesizing, especially the B vitamins, folate, riboflavin, vitamin B12, that is cobalamin, biotin, vitamin K are some of the important vitamins which are produced by different organisms, and they are well known for their functions in energy production, blood coagulation. So the synthesis of these vitamins is very helpful, and it influences lipid metabolism and overall health. And signaling molecules in the great gut-brain axis. There are different metabolites like gamma amino butyric acid, or we commonly call it as GABA, then serotonin precursors. They can act as signaling molecules that influence the gut-brain axis. They also modulate neurotransmitter synthesis, the vagus nerve particularly, the signals, and that's how they are very effective. Now I'll come to the diverse genre of probiotics. Other than Lactobacillus, we have Escherichia, we have Streptococcus, we have Enterococcus, we also have many other species which I will be talking. Propionibacterium and Clostridium are also used as probiotics, where they play an important role in the digestion of the fiber and degradation of dietary fibers. That's how they are so important, and they also produce the short chain fatty acids from the digestion of fiber. And Weissella and Pediococcus produce cytokines, which and other immunity enhancers, and they contribute to the balanced immune system. It may be regulation of the immune system, or it may be strengthening of the immune system. And emerging research on psychobiotics indicates that certain probiotic strains can influence the gut-brain axis, as I was already talking about it, producing neurotransmitter precursors that may affect the mood and cognitive functions, leading to potential applications in mental health. So if your gut is sound, your mind is also sound. Lactobacillus rhamnosus alters the GABA receptor expression significantly, reducing the anxiety and depression-like behavior. So the mice studies have established this, and they have also established the vagus nerve as a key communication channel between gut microbes and the brain. And this study is available in British Journal of Nutrition, where a randomized double-blind placebo-controlled human trial has been reported. I'm especially mentioning this because this is a proper human trial has been conducted with a combination of Lactobacillus helveticus R0052 and Bifidobacterium longum, R0175, which have shown significant reduction in anxiety, depression, and stress scores in healthy volunteers. Now, another term is coming in use, and that is the living therapeutics. Probiotics were earlier uh, considered as generally good for well-being or maintaining the intestinal balance, but not offering specific therapeutic, role. Now the organisms have been designed and they have been targeted to provide therapeutic benefits. That's why they have been referred to as living therapeutics. And these organisms, unlike the conventional drugs, they can sense disease-specific signals, make decisions using genetic circuits, and deliver therapeutic responses locally and dynamically. This enables continuous targeted and adaptive treatment, especially valuable for chronic diseases because they don't go easily. So whenever they reappear, the organisms can sense the biomarkers, respond by producing the therapeutic molecules at the time of need, and act locally, thus reducing the systemic side effects seen with the conventional drugs. Then, as we are all aware that tremendous advances have taken place in genomics and metabolomics and also in metagenomics. So genomic sequencing technologies now enable characterization of probiotic strains, revealing the unique genes, metabolic pathways, and functional traits that explain the strain-specific health effects. So the strain candidate organism, particular strain has to be genomically characterized, and which genes are present, which offer or which enable the particular organism to be effective for imparting the specific benefit to the host. Then metabolomics complements the genomics by identifying bioactive metabolites such as short chain fatty acids or bacteriocins or neuroactive compounds which are produced by the action of different genes. So this combined genomic sequencing and metabolomics, they uh, provide direct evidence of probiotic function and enable functional annotation of probiotic strains. Now, there are new innovative frontiers in probiotic technology, and as we know that engineered organisms are in vogue, they are being used for production of different bioactive principles, and also they are being targeted for uh, inserting or activating the genes of interest, regulating their activity. So these recent advances in synthetic biology have enabled the design of probiotics with tailored functionalities that is enhanced survival in harsh gastrointestinal environment and the ability to produce therapeutic compounds directly in situ, that is directly inside the gut. Unlike traditional genetic engineering, CRISPR system offers sequence-specific targeting that is minimal off-target effects and the possibility of marker-free editing. Marker, as we all know, has been a problem with the regulators because it offers the antibiotic resistance normally, and this resistance is one of the factors for the clearance of the genetically manipulated organisms by the regulators. And that's why this is more essential for clinical applications. So CRISPR modified probiotics can be engineered to improve probiotic fitness, that is stress tolerance, gut persistence, sense environmental cues, pathogen metabolites, nitric oxide, quorum signals, activate therapeutic responses only when needed, and avoid continuous expression, improving safety. Prime editing has been one of the latest developments, and it is a promising tool, next generation CRISPR technology for developing the probiotic candidate bacteria into precision and programmable living medicines that does not require donor DNA templates, allows precise insertions, deletions, and base substitutions, can be performed without antibiotic resistance markers, does not create double-stranded DNA breaks. So researchers have engineered E. coli Nisle, one of the important probiotic strains. Normally, E. coli, common cell E. coli resides in the gut of the healthy individuals, and E. coli Nisle has been one of the candidates from Russia. And now this is being programmed to target DNA sequences associated with antibiotic resistance genes, particularly those present on the plasmids, because they are mobile and the plasmid borne resistance can be transferred. And these organisms, that is the engineered E. coli Nisle, can identify and remove such organisms selectively. This E. coli Nisle colonizes the gut as a normal probiotic. Then engineered CRISPR-Cas3 system recognizes resistance gene sequences, that is normal. Most prevalent is beta lactamase genes in neighboring bacteria, and Cas3 is activated and degrades the resistance bearing plasmids. So the plasmids are removed and pathogenic bacteria lose the antibiotic resistance, and E. coli Nisle itself remains unharmed and retains all the probiotic functions. Then synthetic gene circuits, they process biological inputs using regulatory DNA and proteins to produce specific cellular responses. By combining sensors, regulators, and actuators, microbes can be programmed to detect signals, make decisions, and execute specific responses in a predictable and controllable manner. That is, repressors are used for each gene. Like if suppose there are two genes, gene A and gene B, then gene A produces repressors for gene B, whereas gene B produces repressors for gene A. In the presence of specific inducers, a particular on-off stable state can be achieved. And the first successful demonstration of such a genetic toggle switch, that a logical decision-making circuit could be engineered inside a living cell, has been demonstrated by Gardner and co-workers, and the work has been published in Nature. Now I come to the programmable therapeutic responses. By integrating these synthetic gene circuits, probiotics can be engineered to sense disease-associated signals. That is, what are the markers for inflammation? What are the chemicals which are released during inflammation? So they can be sensed by the probiotic organism. Pathogen metabolites can be sensed, and also hypoxia can be sensed, and because of the respiration by the pathogenic bacteria, and they produce the therapeutic molecules by when they are activated. After sensing only they will produce. So this is at the time of need. The conditional on-demand activity improves precision, safety, and efficacy, distinguishing smart therapeutics from conventional probiotic supplementation. And again, I come to the example of E. coli Nisle with the synthetic circuits to sense intestinal inflammation and produce therapeutic proteins that reduced disease severity in mouse model of inflammatory bowel disease. IBD is a chronic disease, and the model animal studies have been done and published in Nature Biotechnology. Synthetic circuits can also facilitate communication between engineered probiotic strains and convert probiotics from isolated actors into coordinated therapeutic consortia. And there are many examples. One is engineered E. coli with genetic circuits to sense gut inflammation and respond in a controlled manner. Then another study showing engineered bacteria to sense disease states and produce therapeutic molecules in situ. And Kong et al. Also developed synthetic microbial communities that communicate to regular population balance and metabolic output. Quorum sensing is one of the important properties for these organisms. Then there are many critical developmental windows. Gut microbiome is crucial for shaping immune responses and metabolic health. And the gut microbiome undergoes rapid diversification influenced by various factors. So the mode of delivery, type of diet, and environmental exposure are very important. Probiotics can play a significant role in optimizing this early microbial community, thereby enhancing immune system in newborn kids or babies, and then maturation of the immune system and resilience against diseases in their later life when they grow into adults. Specific probiotic strains including Bifidobacterium brevi, Bifidobacterium infantis, and Lactobacillus rhamnosus, they have been identified as particularly beneficial during infancy. This is also believed that some of these organisms are transferred from the mother to the newborn child, and that's how the normal delivery helps in the early establishment of these organisms. And it is also believed that the mother's milk is important because of the bifid factor, and it promotes the growth of bifidobacterial colonization of the newborn babies. These strains not only promote establishment of a healthy gut microbiota, but also enhance immune tolerance, reduce the inflammation, and fortify the gut barrier, which is essential for preventing allergic diseases and other health issues. It has been observed in many reports that allergic diseases are more prevalent in cesarean section born kids as compared to the normal delivery. So the role of microbiota in such allergies in the early stage of the newborn babies and their immune tolerance are very much influenced by the type of microbiota, and a lot of work is being done on, especially on bifidobacteria in this regard. And long-term health and implications, as I was talking about the emerging evidence, which suggests that probiotic intervention can have lasting effects, and these allergies, which come during the later life, which become more apparent, like allergies, asthma, obesity, and metabolic disorders, they can be prevented or reduced if the right kind of microflora is introduced in the early life during the infancy or as early as possible in the newborn baby. This paves the way for future strategies in personalized medicine and also in preventive health care. So probiotics are being engineered, and new innovations in their applications are coming with the advancements. So disease sensing the disease signals, producing the target compounds, and enhancing the efficacy of treatments. A lot of work has been done, and I already talked about the live biotherapeutics, why they are being called as live biotherapeutics, because they are targeted to treat the diseases. And personalized probiotic prescriptions are going to come because the microbiome sequencing is revealing new information on the gut profile of individuals. We can seek information that can align with the personalized medicine, and individual health solutions as it varies from person to person. But there are concerns regarding the GMOs and ethical issues also involved in that. So gene containment strategies to prevent unintended spread beyond their intended target. This has to be taken care of, including kill switches and nutrient dependencies. So regulatory bodies compliance remains a challenge in many systems, and public acceptance and ethical issues as also the acceptance by public, the impression or general narrative in public is against the use of genetically modified organisms. So these societal, ethical, and regulatory questions have to be answered. I personally feel that CRISPR-Cas gene editing may be more acceptable to the regulatory authorities as well as to the public opinion. I wanted to share one more development, that is
Excuse me, sir.
the
Can you
organism I
get?
mentioned.
Excuse me, sir. Sorry to disturb you, sir.
Please.
Can we speed up, sir? Because we are running late.
It's one or two
5
slides
minutes.
more. Only 1 or 2 slides more.
Okay, okay.
A particularly interesting development has been in the field of use of pasteurized Akkermansia muciniphylla, because this degrades the mucin in the hindgut. So unlike traditional probiotics, the heat inactivated form of Akkermansia muciniphylla has been shown to provide beneficial functions for the health benefits, as they need concrete clinical studies, evidence coming from those studies. So I can conclude that the probiotics are evolving with technology. They are becoming more precise, more sensor based, and need based. Even personalized medicine is coming, that is the probiotics which are coming in future may also have this type of application through the personal microbiome measurement, and prescriptions can be done. So thank you very much for your patience and time, and if you permit, I can take some questions if the organizers have some time for that.
Thank
So
you.
I will
Thank you
stop
so much, sir.
sharing my screen, and yeah, Dr. Sahab.
Thank you very much, sir. Actually, you just took a big journey from start to what is going to happen in near future. So it was really encouraging and maybe motivating, and probably the eye-opening for many of the scholars who are sitting here. They will also get many of the research topics to go ahead with that. I'm sorry to disturb you in between, because we are almost half an hour
I
behind the schedule.
appreciate. I can understand
But,
that, and I have submitted my written keynote address also.
Yes, sir.
So
yes,
the
sir.
references are also given there.
Yes, sir.
And those who want to refer, they can see.
Yes, yes. And I think on behalf of Dr. Ameshwar Singh, I also tell to the organizers and the students, we have already published this lecture in this souvenir, and we'll share some of these slides also. And then if you have any queries later on, you can contact your department, or you can directly contact me or Dr. Ameshwar Singh for help. So at the moment, sir, we'll close here. Thank you so much for being with us. And
Thank you for the opportunity, and I really missed it. And once again,
thank you so much, sir.
all the best to you. Thank you.
Thank you so much, sir. And,
So I can leave the platform. Okay.
thank you so much. Bye. So now I leave it to organizers for the next activity, inauguration of the poster session, and then we break for lunch. Okay? And then all our sessions will be in this college, Faculty of Dairy Technology. There are 3 parallel sessions going on, and hence I request everybody to be in the hall exactly at 2:30. We have 50 minutes for the lunch. Ha, here it is over now. Yeah. Yeah. So we are going to the poster place. We are inaugurating poster. Other people can also reach directly. It is near to food court. And then the technical sessions will begin in the Faculty of Dairy Technology. 3 parallel sessions. The program is given. Plus try to reach by 2:30. Thank you very much.
So, thank you so much, sir. So, what we have to do, we have the poster session, inauguration of the poster. So, we have to go to the Faculty of Dairy Technology building, ground floor. There we have the poster inauguration session. And followed by, there will be the lunch, and after that, the session. Thank you.